All right, I think I got it here. Okay. All right, so I wanted to go over chapter 11 on controlling microorganisms because we're going to find that the mode of action for detergents and some of these other antimicrobial solutions in ethylene gas and hydrogen peroxide, they're going to be very similar to how the antibiotics work. So I'm going to go over that first. Sorry, so controlling microorganisms, physical, chemical, and mechanical methods to destroy or reduce undesirable microbes in a given area. So decontamination. So if we're wiping down, those of you who are in lab, if we're wiping down the counters or whatever, we're not sterilizing them, we're you could say we're sanitizing them or we're decontaminating, we're just reducing some of the microbial. So whether we're using detergent to break down the lipids, or we're scrubbing it to mechanically remove a majority of whatever is on the counter, that's one thing. So primary targets are microorganisms capable of causing infection or spoilage. So whether we're talking about bacteria that might spoil something with growing in it, or if we're talking about molds or fungus that would be growing on your fruits and vegetables, and it's June right now, so it would be something that you're going to see. You're going to, whether you're buying something fresh from the farm or it's imported from somewhere, you buy it and then 2 or 3 days later you go to in your refrigerator and grab it and there's mold growing on it or whatever. So theoretically you could just rinse those off, or a really dilute bleach bath, and then rinse them off roll on, put them in the refrigerator will reduce some of that. And we're going to see coming up that they can use radiation to do that. We'll be altering the DNA, and we'll show you that how that is coming up. We'll show you that. So vegetative bacterial cells and endospores. And once again, remember, endospores are extremely hard to kill. The only thing that can really kill them would be autoclaving them under pressure, raising the temperature of steam, from 100 degrees up to 120, 130 degrees. Extremely high temperatures can kill the endospores. We'll see tyndallization coming up, where we can go and we sequentially kill the vegetative cells, and then the spores are left. They're going to turn into vegetative cells, and then we're going to kill them. And we just do this process for about 72 hours, and then we can pretty much be sure we've killed them all. And then we're going to see coming up that theoretically hydrogen peroxide can kill endospores with high enough concentration for enough contact time. And we're going to see that hydrogen peroxide, one of the ways it does is that it keeps bombarding organisms with oxygen. And we know oxygen is toxic, and if the bacteria has no way of dealing with it, it can't live in an oxygen environment. But we're also going to find that even if it can produce enzymes or ways of dealing with the oxygen, we're going to hit it with such high concentrations that it can't deal with it. Sorry guys, my allergies are killing me today. So we have fungal, hyphae, and spores and yeast, protozoan, trophocytes, and cysts. Those are relatively easy to kill. Worms, viruses, actually relatively easy to kill. And then prions, and I mentioned this in lab, if you have me for lab, that for this course, the hardest thing to kill are endospores, but technically prions are actually even harder to kill, and they're not technically alive. They're mutated proteins, but very hard to kill. Okay, what in the world is going on? So this is right out of your book, just kind of showing the physical agents of microbial growth would be here. So heat, radiation, if it's dry, incinerator. So whether you're going to incinerate something in an incinerator, or if you're in lab, we have the plug-in incinerators, or I'm old school. I like the Bunsen burner or a dry oven sterilizing, but that takes extremely long time to do that. Steam under pressure would be the autoclave. If you're canning vegetables, that's you're using a um, pressure cooker, or if you have an Instapot, those are very similar idea. So boiling water, hot water, pasteurization, that's going to kind of reduce the microbial load. Pasteurization. Hot water will kill most organisms. Boiling water will kill most everything that will not kill endospores. All right. And then radiation, we're talking about ionizing radiation, X-ray, cathode or gamma radiation, which is the most powerful. That can literally, I guess it can sterilize things for extreme long period of time. Non-ionizing would be UV, that would be for disinfection. And UV is not really very penetrating, so it has to be something very, very thin. Um, so you could do a UV light under your counter in the bathroom. UV, they sell UV toothbrush disinfecting systems too, or you could just, you know, oil your toothbrush and bleach it, whatever, or just buy a new one. Right, and then chemical agents would be your gases or liquids. And we'll see ethylene oxide coming up here. Liquids, aldehyde, and some of these other ones we can use for disinfection. Mechanical filtration, air and liquids, and filtration is really kind of for liquids. Is if we want to filter something that we really can't heat. So if we're talking about plasma or something, some biological agent we don't want to alter, filtration is probably the best method for that. So disinfection is the destruction or removal of vegetative pathogens, but not bacterial endospores. Usually it's only on inanimate objects. So that would be a counter, I guess, towels or laundry, cutting boards, whatever, something that's not alive or biologically alive. I mean, your skin is theoretically dead, but we wouldn't want to be using it on that either. Sterilization, the complete removal or destruction of all bio or viable microorganisms used on inanimate objects. So this is when we're going to tyndalize something, or we're going to autoclave it under pressure. And if it's sterile or sterilized, that means any conceivable living thing is dead. So once we're done autoclaving things here, I think we do 131 degrees for 20, I think our cycle is 22 minutes, a couple different cycles. But literally when you take anything out of there, it's sterile. It's killed prions, it's killed endospores, and anything in there. And then we send it out and we have it incinerated also. So there's no way that anything from this lab, once it goes into the red biohazard bags and it's autoclaved and sent out, there's no way it's going to be viable, or there's no way it can get out into the environment. Even though we're only using biolevel 1 organisms here, we still want to make sure that that's not leaving this lab. And remember that you're going to wash your hands before lab and after lab, so you're not taking anything, theoretically not taking anything out with you. Please leave your lab coats here, or if you can take your lab coats home, make sure you're washing them. Also, anything you wear in here as far as shoes, you probably want to keep them outside of your house. Yeah, that's probably a good idea. Antisepsis is chemicals applied to the body surfaces to destroy or inhibit vegetative pathogens. If you're going to get some kind of a shot or an injection, they're going to use some kind of degerming agent. Usually, you just simply wiping with an alcohol swab. When I go to the acupuncturist, Dr. Fu always wipes everything down before she flicks that needle into me. So, all right, so relative resistance of microbes. So this is kind of a list of hardest to easiest to kill. And once again, I said prions are technically they're dead. They're proteinaceous infection particles. And usually it's a neurotissue, and it's kind of it causes proteins around it to misform. So it's kind of like the analogy I read was like a snowball going down a hill, right? So it just, as it comes in contact, it causes everything it comes in contact with to um, change form. And bacterial endospores. So like I said, for this course or most other micro courses, endospores are the hardest to kill, and that's, like I said to everyone in lab, you know, if you read a Lysol can or look at Lysol, it'll say kills 99.9% of whatever. The 0.1% is the endospores that can't kill those. Now, moderate resistance, so something that's still theoretically pathogenic and relatively hard to kill, especially in a hospital setting, is Pseudomonas. And that's gram-negative, so the outer membrane prevents penetration of chemical agents. Pseudomonas, when they grow out here in the nutrient broth or the brain heart infused media, over time it gets really slimy. It gets, I don't want to say it looks like a big snot in there, but after a while it looks pretty gross. Mycobacterium tuberculosis, that has a very waxy coating on it due to its mycotic layer, and that's what makes it so difficult for the alpha-2 macrophages in your lungs to destroy it because they can't get past that waxy layer. Streptococcus aureus, so I'm sure you guys have all heard of MRSA, methicillin-resistant Staph aureus. And then once again, remember that when we get into staph, specifically gram-positive staph, it has a lot of virulence factors. It releases a lot of enzymes. It can eat its way through a lot of things. It has a lot of hemolysins, coagulase, can eat its way through almost through cartilaginous tissue. It can just eat its way through the body. Right? And then if it's septic in the blood, it's just a disaster. Protozoan cysts. And when we start talking about the pathogens, I'm sorry, yeah, the pathogens or the eukaryotic parasites, protozoan cysts. So these protozoan are going to be living in your GI tract or wherever it happens to be. And then what they do is they shut off cysts so that they get excreted, and their cysts are hardy enough to live in the environment. They're hoping that some other host will pick them up and propagate that species. Right. So least resistance, most bacterial vegetative cells are relatively easy to kill with any kind of good germicidal cleaner. You know, a lot of the stuff here we have is germicidal, it's fungicidal, virucidal. It kills most everything. I mean, I wouldn't recommend you eat off the counters here, but they are cleaned frequently, so they're relatively clean or degermed, let's put it that way. So fungal spores and hi-fi yeast, relatively easy to kill, and enveloped viruses are easy to kill. You know, even something as simple as UV light will do it, or sunlight. And protozoan trophocytes, that would be the similar to a vegetative bacteria, or the trophocyte would be the feeding stage or the general I don't want to say, yeah, everyday life of a protozoan, but not the cyst form. So resistance of endospores, this is how resistant they are. And just really look at the difference in time and temperature with these. So heat, moist heat, endospores would take 120 degrees, so that's 2 atmospheric pressures. Do that in an autoclave. If it's a vegetative form, it can be done at 80 degrees Celsius, right? So radiation X-rays would be 400 rads or grays now, depending on what book you read. 1/4 of that to kill vegetative forms. Right? Sterilizing gas, ethylene oxide, we'll see that coming up. Endospores, the quantity is much larger. All right, and then sporicidal liquid, glutaldehyde. to kill it, theoretically, you'd have to saturate that for 3 hours, right, or 10 minutes for vegetative forms. So just realize the difference between a vegetative form, which is relatively easy to kill, and endospores. And I talked to you guys in the lab, and it's in the book about the spore formers. They kind of go through binary fission, and then the end spore can more or less goes around the forespore, and then it leaves this coating around it with calcium, very, very hard. And when conditions get much better, there's nutrients and moisture, or whatever, they can come back. And I told you guys in lab that they found these in mummies and these ancient sites, and they can bring them back. So it's just mind-boggling. All right, so these videos I did check, they will run on a regular non-MacBook, non-Apple product. All right, so here's some terminology for your exam. It's all going to be randomly generated, as you guys know. But they'll ask you some, or I'll be asking you some form, or you'll get some version of sterilization. What's in between sterilization, disinfectant, antiseptic, sanitation. Right, so sterilization is a process that destroys all viable microbes, including viruses and endospores. So something sterilized, like we're talking about medical equipment or dental equipment, when they open that, it is completely sterile. All right, but just realize if we're doing like a metal dental instrument, we would sterilize it one way. If we're doing like a endoscope or some kind of camera that's going to be inserted in the body, we want to be able to sterilize that. So we're going to be using ethylene gas or something to sterilize that. We wouldn't want to heat it because we destroy the instrument. Okay. So disinfection is a process to destroy vegetative pathogens, not endospores and inanimate objects. So as far as here, if you're disinfecting if you're cleaning your kitchen floor, you're cleaning your counter or whatever, you're just really you're killing all the vegetative pathogens, but not the endospores. Using on inanimate objects. So, if you have an infant or something like that, you're gonna, I don't know what you're gonna clean with. You're gonna clean with some possibly some bleach solution or something, depending on the surface, whether it's wood or linoleum or whatever. You're gonna clean it, but you just realize you're not going to be getting all the endospores, but it's going to be 99.9% clean. Now, antiseptic. Once again, we all know the antiseptic wipes or whatever. Disinfectants applied directly to exposed body surfaces. So you can use a kind of astringent, face cleanser, alcohol if you're gonna don't give someone a shot or whatever. You're gonna antiseptically disinfect the skin. You want to sanitize it because it's still a biological part of your body. Sanitation or any cleansing technique that mechanically removes microbes. So, I usually say if you go to a hotel or a motel or wherever, you go in your bathroom, it should usually there's a piece of paper over the toilet seat that says sanitized for your protection or sanitized for your comfort or whatever. So just realize that hopefully they wiped it down with some kind of phenolic chemical or bleach or something. So it's relatively clean. I certainly wouldn't recommend eating off it, but you get the idea. Degermination reduces the number of microbes through mechanical means. So we could theoretically say if you spray down a counter and you wipe it, I usually use towels here, but I bleach them. But just it seems to get a lot of the fingerprints and the grease and things off the counter. Oil immersion and stuff, I really want to use a good sturdy cleaner, and then I just tend to wipe it down. Some people will spray it and then smear it with a paper towel. I don't find that to be the best for me, but some people do that. Microbiostatic, I think I spelled that wrong. Stasis, So stasis usually means static or slowing it down. So microbes are temporarily prevented from multiplying, but they're not killed. Static, stasis. So sort of know the difference between whether something is microstatic or microcidal would be to kill it. So static or cidal, there's a difference with those words, right? And not everyone uses these terms properly, but just realize, like I said, static was just going to reduce the ability for that microbe to reproduce, and it can be for a short period of time. It depends, on what's going on. But if it's cidal, it kills everything. Denatures the proteins, kills the endospore, so there's nothing viable that can actually come back. So decontamination, sepsis, aseptic. All right, so in lab we did, hopefully we did aseptic technique where you flamed everything and you kind of flamed the end of your tube, your broth tube, so that you would kill any viable things that might be harbored on the edge of that. And you want to create that vortex to not pull the atmosphere into the tube. So aseptic technique. So technique that prevents the entry of microbes into sterile tissues. So that if you're talking about surgery or doing an injection or whatever, try to use aseptic technique. Antiseptic are chemicals applied to the body surface to destroy and inhibit vegetative pathogens, like I said, an astringent or alcohol or something like that. Disinfection, destruction of vegetative pathogens on inanimate objects. Sanitation, once again, that's just removing microbial load. It's not really sterile, all right? Degermination, basically the same thing. Clean that removes microorganisms and debris from living tissue. So if we're talking about, well, I guess we were talking about gas gangrene or something like Clostridium perfringens. We would want to remove microorganism debris from living tissue. That's actually dead, but we want to debride that area. We want to clean that up. All right, so sterilization, the removal and destruction of all viable microbes by definition. All right, so microbial death is hard to detect. And when you guys were in lab, and I showed you the tubes and I showed you the change in turbidity from the clear broth tube, and then a couple days later, how it was a different color. All right, so we could shine a light through it, and the light wouldn't go all the way through; it would be diffracted. And I said, well, we knew something grew because it changed color, but we don't know if everything in that container is alive. Right? And then we went through the log rate and the stationary rate, the death rate, that curve. So hard to detect microbes often reveal no conspicuous vital signs to begin with. So, I was kidding in lab and I said, well, I can't take its respiration and pulse, right? I can just kind of guess whether it's alive or not. We're assuming it's alive, and then we'd have to plate it. We'd have to, streak plate it or inoculate an auger plate of some sort to see if it was alive. Right? And, we do a streak plate and make sure that we only got one type of organism in there, so there would be no difference in colonial morphology. That it was a pure culture, and then we'd want to aseptically remove one of those colonies, and we'd want to gram stain it and make sure that it was what we said it was. Right. So the E. coli, we want to make sure it's gram negative. Right. So because sometimes things get cross contaminated, that happens. So permanent loss of reproductive capacity, even under optimum growth conditions. So we know if it is something sterile or it's dead, no matter what I do, if I autoclave a broth tube or culture, I autoclave it and I bring it out and I cool it, and then I plate it, put it in the incubator, no matter how, if I even give it like a blood auger plate or put it in the incubator, and every conditions are absolutely perfect. But if there's nothing viable left on there, it can't come back. Right? So some factors that affect the death rate would be the number of microbes. So the microbial load, if there's 2,000 in a tube as opposed to 2 trillion in a tube, it would take more to kill the larger amount of microbes. So nature of the microbes in the population. So with metabolic rates, active cells are younger cells, and when we were doing the gram staining, they said these cultures were only like 24, maybe 48 hours old because we want them to be active. Younger, rapidly dividing tend to die quicker than less active older cells because they're reproducing their peptidoglycan layer and things. It's much more antimicrobial detergent, a much easier way of getting there because it's they're rapidly reproducing. Generally, always a mix of more than one species. All right. So, realize that no matter what you're cleaning, coming across, almost 100% of the time, it's not going to be one single microbe. And I talked to you guys about when we do our skin swabs or even our throat swabs, we can be sure there's going to be more than one microbe. Or if we're talking about the biofilms or whatever's growing on your teeth, like I said, there's over 500 different species of microbes just on your teeth alone. All right, temperature and pH of the environment. So realize that, being cold in general is bacterial static. It tends to slow reproduction, and that's why we refrigerate food, obviously. We freeze food, all right, so it stays for a much longer period of time. But please realize there are microbes that can live in your refrigerator. I've mentioned before, like Listeria, definitely likes to grow on lunch meat, the deli. Or the pH. All right, so if the pH is neutral, all right, most microbes will like that. But if the pH is very, very acidic or very, very basic, it's going to tend to kill most microbes unless it has a niche for that. Like H. pylori, Helicobacter pylori can live in a low pH, acidic environment to some degree. Concentration or dosage of the agent. So if we're using 1% as opposed to 10%, or we're talking about alcohol, like most organisms, like 70% alcohol is usually pretty good. And alcohol is one of those things where the higher the concentration doesn't always make it better. All right, so alcohol is going to start, destroying the biphospholipid layer and the peptidoglycan layer. We'll talk about that. All right, so the mode of action. So what is the mode of action? Is it breaking down the cell wall? Is it breaking down the biphospholipid layer like the polymyxins? Is it stopping denaturing protein? Is it working with PABA or all these different modes of action? Talk about in presence of solvents, organic matter, or inhibitors. All right, so like bleach itself is inactivated by organic matter. So vomit, feces, urine, pretty much, that can inactivate the chlorine and the bleach solution. Right. Sorry. All right. So we're just talking. These are the log rates of whether it is we're sterilizing it, depending on the heat load, time for sterilization. If it's a low microbial load, it wouldn't take as long. If it's a high microbial load, it's going to take much, much longer. And I usually autoclave everything here in small doses. I mean, I could really fill a bag up and do it, but I feel better if I run the autoclave 3 times rather than throwing everything in a great big bag and doing it. And we're talking about here, like vegetative cells are much easier to kill, much less time. Spores are much, much harder. Right. All right. So what are some practical concerns with controlling microbes? All right. And these should seem relatively basic, but just please realize too, it's got to be cost effective. It's got to be available. It's got to be easy to use. Some of the stuff here, when I got here was super strong, and I really didn't really want to be breathing that stuff in. So does the application require sterilization? For everything here, no. But if you're in a hospital setting, you're talking about an operating room or other things like that. So possibly if it requires sterilization, it's a whole different concern. The item is going to be reused like a camera, endoscope. Right? Here. Is it really porous? Like, I don't know. I guess I could say like a cutting board or a wooden spoon or something like that. If that's so porous, you're probably never going to get it sterile unless you autoclave it. Um, but maybe boiling water would suffice. Okay. Is the method suitable for whatever it is? Will the agent penetrate to the necessary extent? Once again, if it's porous, even like some floors are very porous, or they're not smooth, and even glass isn't completely smooth, so it's porous. So we have to be able to get into any kind of crevice or whatever. And your skin too, if you look at um, your skin, the folds in it, or if you're one of those people that uses hand sanitizer all the time, just realize it's alcohol based and you're really drying your skin out, and you're providing, you're getting rid of some of the natural defenses of your skin, the lysosomes and some of the acids, the acidic mantle on your skin. You're getting rid of that. So yeah, your hands are gonna be clean, but you're also wiping out your normal microbiome so that anything um, more pathological has a place to live. We'll talk about that throughout the course here. All right, and then is the method cost and labor efficient, or is it safe? All right, there's a lot of things that are super we could use in our skin that would get it very, very clean, but we wouldn't want that permeating into the skin and getting rid of capillary beds. Right? So here's what I wanted to get through, and then I'm gonna pause it because I've been told that these lectures are too long to focus on, and I guess I get it from your point of view. I could talk forever, but whatever. All right, so anticriminal agents, so the mode of action. So if you see MOA, mode of action. So how is it work? Is it working on, once again, is it working on cell wall, cell membrane, ribosomes? Is it working on metabolism? It's working on sulfidamides. It's working on PABA with folic acid, B9. Then, so the cell wall becomes fragile and cell lysis. Some antimicrobial drugs, like specifically penicillin and cephalosporin, a lot of those, detergents and alcohol, they can start breaking down the cell wall. And then remember that the osmolarity outside of a cell and the osmolar pressure outside of the cell is generally higher than inside the cell. So if we break down that cell wall, it can cause a lot of things to rush into the cell. We don't have that cell wall and that biphospholipid layer that's supposed to control what comes in and out of the cell. It's diminished or partially destroyed, so things will rush in and the cell will burst. All right. Cell membrane loses its integrity. Polymyxins usually work with that. Loses its integrity, like detergents and surfactants. That's how they work. They lower the surface tension. They can affect the inner lipid layer, biphospholipid layers, similar to what our cells are made out of. Okay. So there's surfactants, like most detergents are surfactants, or whatever you're using on your counters is surfactant. It tends to break up the lipids and causes them to break, or causes the cell to break. All right. Cellular targets are physical and chemical agents. So protein and nucleic acid synthesis. So remember, if an organism can't produce protein, nucleic acids, and you guys know, I'm assuming you'll have done chapter 9. I put like 3 videos from, I think it was, I don't know, maybe March. I don't know when I did it, from before on genetics. But remember, we have to have those codons to bring in those amino acids in their certain sequence to produce proteins and producing enzymes. Remember, once again, if an organism can't produce an enzyme, and it can be as little as 1 enzyme, eventually it will die. All right. So prevention of replication, transcription, translation, peptide bond formation, protein synthesis. All right. So formaldehyde, chloraminidazole, UV radiation can do that. All right. And that's how UV radiation, can damage your skin too. It's getting into that nucleus of your epithelial cells and causing that. And that's why, we have all the tanning and trying to protect that genetic information. So some can bind to the DNA irreversibly and prevent transcription and translation. So if it binds to it, it can't replicate, right? It can't make the messenger RNA, and it can't replicate. Proteins disrupt or denature proteins like alcohols, phenols, acids, and heat. Right. Remove the microorganism from its native state. And some can coagulate the proteins. Some agents like metallic ions. So we're talking about copper and some of these other things that they're putting in, on doorknobs and faucets, and they're putting in PICC lines and things now. Attach the active sites and bind up or block the interaction with the correct substrate. So we're actually bonding all the active sites to these enzymes. These enzymatic reactions can't run. So methods of physical control would be heat, moist and dry. And remember, always, moist is always more penetrating than dry. If you're talking about a patient who, wants heat, it wouldn't be an acute, it'd be something chronic, usually muscular. They really want to do moist heat if you can. It's much more penetrating than dry heat. It's deeper. Cold temperature, we really can slow down static. We can slow down the reproductive rate, and very similar to humans, more or less. When it's super cold out, people get a little more sluggish. When it's warm out, people are a little bit more active. When it's too warm, though, that can be a problem too. Desiccation, so that would be drying it out. And if you ever buy electronics or a lot of things now, even shoes or whatever, you're gonna have those little packets, and they're designed to pull the moisture out of the air. So, by the way, don't throw those out, all right? Because if organisms eat them, it can destroy their intestines. And just keep them in a Ziploc bag, because if you drop your phone and something wet or whatever, you can throw it in a bag with those. It'll pull the moisture out. Some people use rice or whatever, but you want to keep those. Put your cell phone in there, and it'll wick all that moisture away. All right. Radiation, we're going to see how that's going to start affecting the hydrogen bonds of the DNA helix, depending on whether it's eukaryotic or prokaryotic. It's going to really start affecting the bonds of the genetic information. And then filtration, I said before, we really want to use that for, you can use it for air, or we're going to really use it for any kind of a body fluid or something that we really don't want to heat or chemically alter. And moist heat, lower temperatures and shorter exposure time because it's much more penetrating. Coagulation and denaturing of proteins. All right. And like I said, when you start thinking about denaturing proteins, you could be talking about large proteins, like if you don't do it anymore, but if you were to perm your hair, all right, or chemically relax it or chemically curl it, you're going to start denaturing the protein. You start messing with the sulfide bonds. But for biologically, just remember we're going to be affecting the enzymes, all right? Which, remember, enzymes, they decrease the activation energy. So these reactions can happen at a much faster rate, or lowers the energy of activation for these. So they can happen much faster. These reactions would happen, but they'd happen so slowly that it wouldn't be viable with life. It would just, you wouldn't be, yeah, it wouldn't be life sustaining. And then dry heat, moderate to high temperature. So it's always a higher temperature. It's not as penetrating. That can dehydrate things, alters protein structure, or an incinerator. Now, an incinerator theoretically will kill everything, right? That'll just work. Just like if you're using the Bunsen burner here or those electric incinerators. It's a dry heat, but it's a higher temperature. It's not as penetrating. And this tends to dry things out. And usually most anything but endospores can't live without some type of moisture. Right? It does alter the protein structure. Right? So there's just a difference in the time it would take with moist heat, the temperatures, and dry heat. So moist heat takes a lot less time. So if you're steaming something or you're tyndalizing it, it takes a little less time. Right? I'll do this slide and then I'll end it. So thermal depth measurements. Bacterial endospores are the most resistant, usually require temperatures above boiling. That's why we have to put it under pressure. We have to, water boils at 100 degrees. We need it to be at 120 or 130 degrees. So to put it under a lot of atmospheric pressure to cause the water to boil at a higher temperature. And then if it boils at a higher temperature, the steam is obviously going to be higher temperature. All right, so thermal depth time. You'll probably get some version of this question too. Thermal depth time is the shortest length of time required to kill all microbes at a specific temperature. So we could set up an experiment here where I had all of you with your hot plates and you had, I don't know, we could do one, hot plate at 75 degrees, one at 80, one at 90, one at 100. All right. And then, I'm sorry, no, that would do thermal depth point. So we had everyone have their water at, let's say, 100 degrees. All right. And then we would have somebody do their microbe for 4 minutes, one at 5 minutes, one at 6 minutes, one at 7 minutes, and then we'd come back and we'd plate all those and see at what temperature they stopped growing. So then everything subsequently after that would be the thermal death time. Anything after that would be dead at that point in time. But thermal death point, we do 10 minutes and then we change the temperature and then we come back and we plate it all. So we would say, well, the lowest temperature required to kill all microbes in a sample in 10 minutes. So we do one beaker at, and we can make it up, 95, one beaker at 100 degrees, one at 105, one at 110, one at 120, everything for 10 minutes. Then we'd come back and we'd plate from that broth that we did, and we'd see which lowest temperature required to kill all microbes in a sample at 10 minutes. So if at 85 or 90 degrees they lived, or at 95 they didn't live, so at 95 they wouldn't live, at 100 they wouldn't live, 120 they wouldn't live. All right. Okay, so let me pause this. This can't possibly be any harder. Where in the world is